How are you guys doing?
Pretty good.
You ready for Thanksgiving?
Yeah, for sure.
Everybody's like, please make it stop. I'm so tired of Zoom. I had a meeting earlier today about the undergraduate colloquium, which is in the spring, and it is not likely that we will be holding colloquium in person. So we were discussing options for doing it virtually. And one of the committee members was like, by April, is anybody going to want to have colloquium virtually? I'm like, I don't know, but you just, we didn't have it last year because after it went virtual, only like 3 people wanted to do it. And I don't know that we want to go 2 years without doing it. So we'll see how it goes. Katie, you already have your Christmas tree up.
Yeah.
When do you guys typically do that?
Usually we wait till like the day after Thanksgiving, but I guess my mom wanted it up early this year.
Yeah, I usually do mine after Thanksgiving also. Keaton, is your beard getting longer?
Aw.
Yes, it is. I left my trimmer at school, so just growing it
Oh.
out back.
Oh, okay. All right. Yeah, I thought you were maybe looking a little scruffier than I recall.
Yeah, it's kind of growing on me, but we'll see.
Where is home?
Lawson, Missouri.
Oh, so you could come back and get it if you wanted.
Well, I'm technically not allowed on campus right now, so.
Oh, you're in quarantine. Okay, gotcha. So, oh well. So I guess by spring you'll just be like Grizzly Adams or something.
Yeah, might dye it white, be Santa Claus. I don't know.
Okay. Actually, I know a guy who does Santa duty in the winter, and he's a big guy. I have a bicycle with him. He's a big guy, and he's got a big beard, and he dyes it white every year.
My dad actually does Santa Claus gigs because he's like 6'6", and he always grows a big white beard. So it's fun.
Yeah, that's weird that I now have associations with 2 professional Santas. That's bizarre. Yeah. The guy that I know that I ride with, his name is Bill Quassa, and he goes by the Santa Quass because his last name is Quassa.
That's funny.
I don't know. Do Santa professional Santa cosplayers do they like all know one another?
I don't know. I'm sure there's like a community, like a Facebook group out there somewhere.
Yeah. Huh. I'm gonna have to ask Bill about that next time I see him. All right, enough about Santa. How's everybody else doing? All right, last day, and then we get to take a week off. So we were talking last time about this case study and the chapter from that book that I mentioned to you, and so I just want to continue with that. But before we continue with that, do you guys have any questions about what we were talking about last time? Dr. Judd? You look like you're about to say something.
Oh, no, I'm trying to make sure I have everyone's lab data all sorted out in a way that you and I can share. Yeah, so background stuff.
Okay, background stuff. Okay. All right. Then if there are no questions, we'll just press on. So we were in the process of talking about how Brody III was in Oregon. He was catching these snakes for a quantitative genetics project. And as he was doing this, he noticed that some species of snakes were eating, you know, slugs and worms and things like this, and other species of snakes were actually eating these newts that he knew to be toxic. And he knew that these snakes were toxic. Why?
Didn't he know through that like story about like the newts in the coffee pot?
Yeah, so his dad, who is right here, and so if you have any doubts as to whether genetics is a thing, look at Butch Jr. and Butch the Third and the grandson. The grandson is not showing the Brody morphotype yet, but it's clear that Big Butch and Little Butch are genetically related to one another. But his father, when he was an undergraduate, he went to a professor at the school where he was at, and he said, I want to do some undergraduate research at a time when undergraduate research really wasn't much of a thing. And his professor said, oh, well, I read about this in the paper, and why don't you go and see what the deal is with this? And so Tessa, tell us the coffee pot and the fisherman story.
So weren't there like 3 hunters and they were like found dead at this campsite and there weren't any like injuries or anything, but they found a newt in a coffee pot. So they decided, I think Brody skin of the newts by like grinding them up and then giving them to field mice, and like the field mice died within a couple minutes of eating the skin.
Yeah, so these hunters had presumably scooped up some water for coffee from the neighboring stream. Didn't realize that in the process of scooping it up, they scooped up a newt, boiled their coffee and drank their coffee, and presumably drank up a bunch of tetrodotoxin, what we now know is tetrodotoxin. They didn't have a name for it at the time. And so what he did was he went and collected a bunch of newts, and he fed them to lots of different organisms. This is one of this is the title page of the paper from Copia in 1968. This is table 1 from that publication, basically showing the wide variety of animals that he fed pieces of newt skin to and the time that it required them to succumb to the toxin. And he had figures. He fed it to terrestrial organisms, to aquatic organisms, and if you give them more skin, they die faster. And so he knew that there was a toxin. Bob Storm was who put them on this project. Or I guess Bob Storm was his master's advisor, not the guy who put him on the project. But Ken Walker was the guy who actually put him onto the project to begin with. And so he found that these newts were actually toxic, and they were toxic enough that if you drank some coffee from a boiled newt, that it would probably be enough to kill you. And we've learned a lot about tetrodotoxin since that time, but this was the beginning of all of this. And because this was Butch Brody III's father, he had heard this story from his childhood, and they're actually from Oregon. So Little Butch's grandmother lives in Oregon, and so this was familiar to him. So this whole thing brought up a bunch of questions in Butch Brody's mind. You know, what are the ecological forces that are pushing snakes to eat poisonous prey? Was resistance accidental byproduct of garter snake physiology, or did these snakes evolve resistance in direct response to eating poisonous newts? In other words, are the newts basically imposing selection pressure on the snakes? What are the mechanisms that could enable garter snakes to resist a toxin that killed every other vertebrate predator? And were resistant snakes the evolutionary cause of the extreme toxin in the newts, indicating that as the newts get more toxic, they make the snakes more resistant, and as the snakes get more resistant, do they then make the newts more toxic over evolutionary time? So this first question, what ecological forces push snakes to eat poisonous prey, is easiest to answer when we think about this particular group of snakes. These snakes are in the genus Thamnophis, and this is a group of snakes that is well known for being dietary specialists. And so this is a paper by Stephen Arnold. Stephen Arnold was Butch Brodie III's graduate advisor when he was a PhD student at the University of Chicago. And he had done some work back in the 70s and early 80s looking at chemosensory behavior in snakes. And so what he would do is he would swab different kinds of prey items and then present that swab to a snake. And a snake uses its forked tongue to gather scent particles and then brings it into an organ on the roof of the mouth called the vomeronasal organ. And so it uses the tongue to improve its sense of smell. And one of the things that happens is when a snake is interested in something, the tongue flick rate increases drastically. And so he basically used this to assess whether or not snakes were interested in different scents from different types of prey. And he looked at coastal populations and he looked at inland populations, and he saw that they had very different preferences for the types of olfactory stimuli. And so in this case, this is slug eating. So the coastal snakes tended to respond better to slugs than the inland snakes do. And presumably that's due to the fact that these coastal populations live in a moisture environment where slugs make up more of the diet. And he found that these differences were not only very different between the coastal populations and the inland populations, but when we look at crosses of the coastal and inland populations, you start to get more of the inland, the genes that determine the response in the inland kind of swamp the genes that are responsible for the coastal population's preference. And so one of the things that might force a snake to eat a newt is if it starts out as a dietary specialist to begin with. And so when you look at snakes in the genus Thamnophis, some of them are dietary generalists, meaning that they'll eat anything. They'll eat insects, they'll eat frogs, they'll eat salamanders, they'll eat slugs. But a lot of the snakes in this genus are actually dietary specialists in that they only eat slugs, or they only eat frogs, or they only eat fish, for example. And so one of the reasons that this might have happened is that if the snakes are dietary specialists on the newt might then have genetic variation for tetrodotoxin toxicity, and the snakes start exerting selection pressure onto the newts so that they become more toxic over evolutionary time. But then that has the potential to exert selection pressure on the snakes such that only those snakes that are resistant can continue eating the newts. And they could go and eat something else, except that they already have this dietary specialization that is well known among members of the genus Thamnophis. And so here's a picture by Brodie of a garter snake, Thamnophis ortalis, consuming an adult newt, an adult rough-skinned newt. So as you go through this chapter, one of the things I wanted you to pay attention to that I told you about on Wednesday is to think about what does it take to be a scientist? You guys are at the beginning of your careers of becoming scientists. It's good to know what are the characteristics of a scientist that is kind of illustrated by this story.
Ask questions.
Okay, you have to be infinitely curious. You have to be curious about the natural world around you. You have to be curious enough that you go to Oregon during the rainy season. And if you've ever been in Oregon during the rainy season, it's really rainy there at certain times of the year. And you have to be curious enough that you're willing to put up with being damp and moldy for months at a time in order to get answers. What other kind of characteristics are there revealed by this story?
You have to be dedicated. I mean, several generations of the.
Sorry, Tessa, I lost the last end of that.
Sorry, my internet keeps cutting in and out. You have to be dedicated. The Brodie family dedicated multiple generations to studying.
Yeah, Yeah, so in this case, they've been studying this system since the 1960s. It languished for a while there. They picked it up once again in the 1980s, late 80s, but ever since the 1980s, this has been one of the main components of their research program in both the lab of the father and in the lab of the son. What else? As they got further and further into this project, what happened in terms of who they worked with?
They have a lot of connections with other people in different fields.
Yeah, over time, as they got further and further into this, they recognized that there were some things that they needed to be able to do in order to answer the kinds of questions that they had. And they recognized that they didn't have the expertise to do those things. And so what you do when you don't have expertise is you go find other people who do have expertise, and you rely on those people to teach you the things that you don't know and to collaborate with you so that you can get the questions answered that you're having. And so I think one of the impressions that you guys might get about Dr. Judd and I is that we know everything about everything because we know quite a lot about ecology. But the fact of the matter is there's a lot of stuff that we don't know anything about because we aren't really formally trained in those areas. And so being ignorant is actually just kind of part of the human condition. And simply because you're an expert in one area doesn't mean that you know anything about another area. So another thing about being a scientist is being humble enough to recognize that you don't know everything about everything, and that there are areas of knowledge that you are ignorant of. And if you need to know those things, you need to figure out ways of becoming educated about those things. Dr. Judd, you don't know everything about everything, do you? Can't hear you. Anyway.
I was going to reflect on your point there.
Oh, yeah. I still can't hear you. There you are.
Is that better?
Yes, that is better.
Okay. I just wanted to reflect with you all for a moment on this point that Dr. Klinsky was making. You all aren't just learning biology content, you're learning how to learn. And that's one of the things that makes ignorance kind of a positive thing when you recognize it, because as you develop the skills on how to learn things, how to find things, how to investigate, you're adding capacity for yourself to learn in the future and the capacity to discover things.
And sometimes students are put off by my use of the term ignorant. I am ignorant of genomic biology. That doesn't mean that I am not capable of learning that field if I set my mind to it or anything else. It just means that I've never gone to the trouble of learning that. And I'm ignorant about all kinds of things. I don't view the term ignorant to be a pejorative term. It's just a statement about what your state of being is at any particular moment about a particular area of knowledge. I'm super ignorant about French. I cannot speak it. I cannot understand it when other people speak it. And I've made zero efforts to do anything about that particular area of ignorance. So don't think that if we use that term that there's something negative about it. It's just a state of being that presumably is rectifiable if one wants it to be. The couple of figures ago, there were a series of questions, and these questions represented, getting back to Paula's point, these questions represent an increasing kind of level of depth as you begin to think about this system. And so one of the things that we hope that you're also learning as you go through this is that you learn how to ask questions, and that the questions that you ask are questions that get to the heart of what the issues are about the biology that you're learning about. So other questions that you might have about this is, how does tetrodotoxin act to paralyze snakes and other vertebrates? How do resistant snakes overcome this? And is there a cost to this resistance? And in the chapter, they talk about all of these things. Did you guys, so let's answer some of these. What did they ultimately find was the action of tetrodotoxin? how does tetrodotoxin paralyze snakes and other vertebrates?
Someone new. What did you guys find?
It is in the chapter.
Didn't they say it like affects the muscle and like nerve channels in the snakes?
Yeah, so it blocks sodium channels, and sodium channels are necessary for nerve transmission in particular. Muscle contraction is due to calcium channels, which it doesn't block, but it does block the nerve impulses because it blocks sodium channels. So how do resistant snakes overcome this blocking of sodium channels? Somebody other than Sydney.
They had different amino acids that like were substituted.
Yeah, and so there are mutations in the gene that is used to build, that specifies the order of amino acids in the sodium channel. The sodium channel is a transmembrane protein, so it's specified by the code in DNA. And there's a mutation, there's a series of mutations, but there's one mutation in particular that allows snakes to create sodium channels that don't bind to tetrodotoxin as tightly. And as a result, it doesn't tend to block the sodium channel. And did he mention in the chapter whether or not there was a cost to this resistance? Is there a trade-off such that if you're really resistant to tetrodotoxin, you suffer some other cost to some other aspect of your phenotype? Grant, is there a cost to this resistance? Grant, can you hear me? Audrey.
I mean, I think there might be a small cost just to their performance biologically in general, but I don't remember there being one specific large trade-off that they had.
Yeah, can anybody help us out here? Well, maybe it doesn't mention this in the chapter. Well, anyway, there is a cost, and we'll see it when we look at the regression stuff. It turns out that the more resistant that you are, the slower you are when you're not being affected by tetrodotoxin. So there is this trade-off. The idea of trade-offs is really important in ecology. Some of the groups in lab are studying trade-offs. So for example, when a plant grows, it can put its energy into growth, or it can put its energy into reproduction. Any energy that it's put into growth, it can't put into reproduction because you only have a certain amount of energy to go around. And so that represents a trade-off. You can either grow big, but then that takes energy away from reproduction, or you can grow small, which maybe then allows you to put more energy into reproduction. We've also talked about the size number trade-off in lab, having to do with seed size versus seed number. all of those kinds of things are trade-offs. So they did mention this. I'm 99.9% sure they mentioned the geographic mosaic theory of coevolution. What is that? Somebody try and describe that for us. Cody, give it a shot. Hunter, what is the geographic mosaic theory of coevolution?
I have no idea, to be honest.
Okay, call in somebody else to help you out.
I guess Katie.
So I understood that it's like basically that evolution, like in like for stuff like this, happens in more of a specific area based on that like species and what exactly they have to eat, instead of like on a broader scale to the whole species.
Okay, that's not really quite what it's about. Somebody else, give it a shot. So these snakes are resistant and the newts are toxic. Are the snakes resistant to the same degree of resistance everywhere?
No, I think doesn't it depend on the geographic location?
Yeah, so it depends on their geographic location. So in some places, the newts are not very toxic, and so the snakes are what?
They're not as resistant because they don't have to be.
Yep, and so in other places where the newts are more toxic, the snakes are
more resistant.
Yeah, and so you have this geographic pattern where in some places, newts are not very toxic and snakes are not very resistant. In other places, newts are very toxic and snakes are very resistant. And so what happens in any given population of snakes and newts has to do with things going on in that population. It's not this generalized response at a species level, it's more of a population response at the population level. And so what that ultimately implies is that in some cases, newts didn't stumble upon producing tetrodotoxin as a way of defending themselves. And as a result of not stumbling upon that mutation, then those snakes never became resistant to tetrodotoxin because the newts just don't produce it in large enough quantities to be harmful to the snakes. And so the last paper that we look at will actually look at this geographic mosaic theory of coevolution in the context of this particular study. And I think that's that for that.
We talked about analogies this week in lab, and I want you all to see that the analogy here is built right into the term. Were you going to hit on that, Paul?
I wasn't going to hit on that.
I think it's worth it here. Do you guys know what a mosaic is?
Isn't it like a collage? I know they have usually like a stained glass mosaic is like a collage of different glasses.
Yeah, there's lots of little pieces and they're all packed together usually. Often, historically, they're made out of tile, bits and pieces of tile with different shapes and colors that you can put together to make a picture. So you can think of the different geographic areas where you have different resistance levels and toxicity levels as each being a different tile in a mosaic. And the amount of resistance and the amount of toxicity is different from tile to tile. So that's what we mean by a geographic mosaic across a landscape, a real landscape.
Yeah, in this case, we are talking about a landscape rather than a metaphorical or theoretical landscape. All right, questions about any of that? Hopefully that chapter is written in such a way that it helps you attack the literature that you're going to be attacking later on in this case study. The first part of this case study, you're going to get, kind of the theoretical foundations, but then the last half of this case study is all done through the literature, through the primary literature. So if you look on Moodle, The first paper that you have on the Moodle site, when is it? I don't know the date, actually. I don't have Moodle up in front of me. But the first paper in that sequence, you need to have a literature summary of. And that's the first of a series of papers that we'll read that will take us all the way through the last day of classes. And so each day we're going to be reading a paper, and we're going to be discussing that paper in class. And that's how we're going to learn the latter half of this case material, is through the primary literature. As with all these case studies, what we begin doing is we introduce you to the statistical techniques that they use in the papers. And in all of these papers, a repeatedly used technique is regression analysis. We've already introduced you to regression analysis in lab when we talked about statistics, but I want to go back through it because I also want to cover some things that we didn't cover with you in lab. So the first part of this, I'm going to move through pretty quickly. Stop me if I'm going too quickly so that we can get to some of the stuff at the end. So regression is this idea that we're relating one continuous variable to another continuous variable. And in this case, we usually think of it as the X variable causes the Y variable. And so if we're going to make a graph of this, we have some X variable, we have some Y variable, and the assumption in regression is that there's a causal relationship such that if the researcher alters X, it will lead to a correlated response in Y. And that's a very strict definition of regression because correlation is the looser definition of this. It's just the degree to which two variables vary with one another. And it doesn't imply any sort of causation. Generally, regression in the strictest sense implies that X causes Y or X predicts Y. We generally have this in the form of a line, Y equals A plus BX. A is the Y-intercept and B is the slope. X obviously is the X variable and Y is the Y variable. We talked to you about two types of regression: ordinary least squares regression, which is regression like I just described, where the independent variable is under the control of the researcher. So the researcher can set what X is. And then the question is, how does Y respond to changes in X? So for any given value of X, the Y's are independently and normally distributed around the average response at that X value. The example that we'll work through is actually one of these where the researcher actually set what the X value was. This actually turns out to be not the way most scientists do regression. So there is another thing called a model 2 regression, where both the X variable and the Y variable are random variables. And so you go out and you grab a snake and it has a certain resistance value, and it also then has a certain sprint speed value. And those aren't set by the researcher, those are ultimately set by the snakes themselves. And so they vary randomly. And you do a different method of regression when you have that type of situation, or you're supposed to do a different method of regression when you have that type of situation. What you find when you get to the literature is that most people don't do a different form of regression when they have this situation. But they could, they just don't usually. So let's look at the example that we covered in lab. We have these flower beetles that are under different conditions of relative humidity, and the relative humidity is set by the investigator. They weighed the beetles in 9 groups of 25 beetles apiece, and they calculated the weight loss or gain after 6 days of being at a certain amount of relative humidity. So the independent variable, which is the X variable, is relative humidity, and the dependent variable, which is the thing that is presumably responding to relative humidity, is weight loss. And these are what the data look like. I do question a little bit that these are actually set by the researcher, because why did the researcher choose 12% humidity and then 29.5% humidity? Perhaps those were just the values that they could achieve consistently in the lab. But anyway, what you notice about this is that at low relative humidity, the weight loss is higher, and at higher relative humidity, the weight loss is lower. And we can show that just by plotting weight loss against relative humidity. Now, the average weight loss for this particular experiment is 6.022 milligrams. Sorry, not grams, but milligrams. And so one of the things that you could assume at the beginning is that these two variables are not related to one another. And if these two variables are not related to one another, what you would expect is just a horizontal line at 6.022. But of course, when you put a horizontal line through these data, what do you notice about this?
It's not
It's
really.
not the.
Somebody other
Is
than Tessa and Connor. Sorry, Tessa and Connor, somebody else.
That line doesn't really fit the data. Like there's a lot above and a lot below.
That is
And
a terrible fit to the data, isn't it? Yeah, exactly. And so the distance between the line and the data points themselves, these are referred to as residuals. So whenever we use the term residual, it's the term that we use for the difference between the individual data points and the mean. And in this case, the mean is just 6.022. And so what we see is that at some parts of this graph, the residuals are quite large. At other parts of the graph, the residuals are also quite large. And here in the middle, the residuals are pretty close to the line. Well, what this indicates is that this shouldn't be a flat line. It should be a line that varies as x varies. And so if you calculate the regression, you end up with a line that has this equation where minus 0.0532 is the slope value. This is the y-intercept, 8.7, way up here close to 9. And when you draw that line through the data points, you find that the residual values, the distance between the line and the data point, are now much smaller. And that's what regression analysis does, is it attempts to find the slope of the line that reduces the total amount of residual variation. These residuals are just the sums of the squared deviations of the data point from the line. And in this case, the line is acting like an average. It's just an average of the y values as x varies. So that's another way of thinking of this line. This line is the running average of y as x changes. And so in that way, it's very analogous to an average in an analysis of variance, and then you have variation around that sample average. And so similar thing here is, here's the residual value between 9 and 8.7. That value is positive, this value is slightly positive, this one is negative, that one is negative. If you sum up all of these residual values, they will sum to 0, which is why when we are working with residuals, we always square them so that they all add up to something. And what regression is trying to do is to reduce that total amount of residual variation around the line. We're not going to teach you the mathematics of having to do this, but we want you to understand in concept what it is doing. So now these residuals are smaller. You'll notice also that in this figure, we have an R-square value of 0.9745, and that just relates to how much spread there is around the line. And it's actually well, let me think. Yeah, I'm not going to say that because I haven't checked myself. All right. So when you look at R square, if every point was on the line such that X tells you 100% tells you about 100% of the variation in Y, then you have an R square of 1. Because what an R square value does is it basically tells you how well does X explain variation in Y. So to what degree does variation in X explain variation in Y? And in this case, variation in X explains 100% of the variation in Y. Here the data are more spread around the line. And as a result, knowing something about X tells you something about Y, but it doesn't tell you everything there is to know about Y. And so the R square value here is about 0.9. Here, the data are spread even further around the line. So knowing something about X still tells you something about Y, but it doesn't tell you as much about Y as it does in these other two situations. And eventually you get to the point where knowing something about X stops telling you anything about Y. And so this is still a significant correlation, but once again, X is not a very good descriptor of Y in this case. And so that's what R square is telling you about. So when you report regressions, we would like to see not only the equation for the line, and we also want to see what the p-value is that is associated with whether or not that slope is significantly different from a slope of 0, but we also want to know things like R square. How well does variation in X describe variation in Y? And the reason that this is important is because you get situations like this, where you have a huge data set, 336 pairs of, or 337 pairs of birds, and the p-value on this is a very highly significant, statistically significant regression, but you'll notice that the R square value is 0.09. That means that variation in male tail length, so this is looking at female tail length versus male tail length in a group of birds, barn swallows. And so variation in male tail length does tell you something about variation in female tail length, but male tail length only describes about 9% of the variation in female tail length. So while the slope is significantly different from 0, from a slope of 0, the regression itself isn't particularly informative. X doesn't really inform you very well about what is going on with Y. And so the nice thing would be to have a highly significant regression where X tells you a lot about Y. What this means is that there's a lot more going on with female tail length than just male tail length in this particular system. We use regression to calculate heritabilities. And so once again, same application of the technique. We have mid-parent height and average offspring height. Presumably the genes in parents determine the phenotype of offspring. And so this is once again a causal relationship. And in heritability, and we'll get into this once again more in detail later on in the case study, heritability is the value of the slope. So in this case, heritability is 0.9165, which is a quite high heritability estimate. So to drive home this point further, there's a shoe data size data set that I pulled off the web some years ago. It's 408 data points. It has the shoe size and height of men and women. And one of the questions that you might have is, do men and women have different shoe sizes? And you would typically examine this with something like an analysis of variance. And when you take these data and you put it to an analysis of variances, what you find is that there's a highly significant difference between males and females in terms of shoe size. P-value is 2 times 10 to the minus 16th, which is the lowest p-value that R will calculate. And you can see that these differences are very big. But why are these differences so big? What's the other difference that isn't in this data set if we're just looking at shoe size that would be correlated with this difference in shoe size?
Height, probably.
Yeah, height.
Was it?
So, yeah, go ahead.
Oh, were you meaning to ask like that question to be answered?
Yeah, yeah.
Oh.
Every question I ask, I'm asking to be answered.
Okay.
You guys haven't figured that out yet? Yeah, so part of this difference in shoe size is just difference in height. Men are taller than women, so men also have bigger shoe size than women. If we look at the relationship between shoe size and height for men and women, the women are in red, the men are in blue, we see that there is a correlation. This regression is significant, it's highly significant, and the R square value, knowing something about height, actually tells you quite a lot about shoe size, 0.75. We can jitter the data so that you can see the individual data points, and you can see that right in this area is where there's a lot of overlap in the height of men and women, but there are a bunch of women who are shorter than the shortest man, and there are a number of men who are taller than the tallest woman. And so one of the things that the Brodies do occasionally is they actually look at residuals. instead of looking at the actual values of things, they'll actually look at the residual. How far is this dot from the average? How far is this data point from the average? How far is this female from the average? If you're below the line, you have a below average shoe size for your height. If you're above the line, you have larger feet for your height. So calculating the residuals gives you a measure of shoe size that's kind of been corrected for height. And when we do this with the shoe size data, we actually find that residual shoe size is much closer in males and females because you have removed the size of height from the equation. Now, there's still a significant difference between male and female height, but this is one of the ways that you can use residual variation to statistically remove another variable that is correlated with the variable that you're really interested in. So when you see them referring to residual resistance and things like this, this is what they're talking about. This is a figure from one of their papers where they look at this trade-off that we were talking about before, and it shows you the baseline speed before they inject a snake with tetrodotoxin and the post-injection speed, which is the speed after you get injected with tetrodotoxin. The dotted lines are what you would predict would be the speed if the baseline speed was the same as the post-injection speed, and the solid line is the fitted data through the cloud of data points. And what I want to do is show you how you can use regression to get at this idea of a trade-off. So if we have baseline speed and post-injection speed, if there is no effect of tetrodotoxin, what would you expect this line to be like? If tetrodotoxin has no effect on the speed of snakes, what would you expect the post-injection speed to be relative to the baseline speed before injection with tetrodotoxin?
The same.
You would expect it to be the same. Who was that?
It's me, Ryan.
Oh, hey, Ryan. Yeah, so you would expect this to be a one-to-one line. If you slithered at 5 centimeters per second before injection, you should expect to be slithering at 5 centimeters per second after injection. You're a slow snake, but you're no slower after injection than you were before. If you're a fast snake, then you would expect to have the same speed if there is no effect of tetrodotoxin. Now, if there is an effect of tetrodotoxin and every snake is affected by tetrodotoxin equally, what would you expect to find happening to this line?
The slope would decrease.
Would the slope decrease or would something else change? If everybody is affected equally by the presence of tetrodotoxin. Take a second shot at it, Katie.
Like, do you mean like if they like all ended up at the same speed afterwards, or like it decreases the same amount?
If it decreased to the same amount, so let's say that every snake was 20% slower than it was before, what would you expect to happen with that line? Would the slope change or would something else change about the line?
I don't know.
Somebody help her out.
Wouldn't the numbers on the side change?
Well, the numbers wouldn't change, just the position of the line would change.
Ask yourself, what are the two parameters of a line? You will have identified slope. What's the other one?
Y-intercept.
Yeah, so Connor, the y-intercept will change, but the slope will not. And so this is where everybody is affected by tetrodotoxin equally. If you're a fast snake, you're still fast, but you're a little slower than you were before. If you're a slow snake, you're still slow, you're just a little slower than you were before. And so this is a line that you would expect if there was an effect of tetrodotoxin, but there wasn't a trade-off. Snakes that are fast are not affected by tetrodotoxin any differently than snakes that are slow are. When you get the data, though, the data end up looking like this. What does that look like has happened? Has the slope changed, or has the y-intercept changed, or have both of them changed?
both.
Yeah, it looks like both the slope has changed and the y-intercept has changed. So what we can do is we can get the average baseline speed and the average post-injection speed, and we can plot a line through that which represents the line that we would expect if there wasn't any trade-off, if all individuals are affected equally. And so you see that this line is parallel to the red line and to the black line, in that snakes that are fast are affected to the same degree that snakes that are slow. But this is a line that is based on passing a 1-to-1 line, a 45-degree line, through this average of post-injection speed and baseline speed. But when we go and actually do a regression that fits the model, that fits the data best, we actually find that that data has a slope that is lower than 1. And what that means is that snakes that are fast tend to be affected more by tetrodotoxin than snakes that are slow, which tend to be affected less by tetrodotoxin. So when you get rid of all of that other stuff, you get a line, a couple of lines that look like this. And what determines the trade-off is that slope lower than a slope of 1? If it's lower than a slope of 1, faster snakes are being affected more by tetrodotoxin than slower snakes are. And that's what these figures look like. And you can see in every one of these situations, the slope of the line of the observed data is different from the dotted line, which is the slope of 1. So when you get to this paper, one of the things that I'm going to tell you to do is to come back to this PowerPoint presentation and revisit these slides so that you can remind yourself what exactly is happening in all of this. Well, what would we expect if tetrodotoxin had no effect? What would we expect if tetrodotoxin has an effect, but the effect size is the same for everybody? What's some actual data look like? Well, this is what the tetrodotoxin effect that is the same for everybody would look like, but this is actually what the data show, and that change in slope is what constitutes the trade-off, which then gets you to this figure, which is the figure that is in the papers. Okay? So, but once again, this is just an application of this general idea in statistics of regression, relating one variable to another variable. That was a ton of information in a very short period of time. What questions do you have about any of that? Nobody? Was that all fairly clear, or was it just so opaque that you don't know where to begin? Which is also okay. Let's clarify it now, though. All right, then. I forgot to put these on the Moodle site, but I'll upload these regression slides onto the Moodle site so you can go back through them if you need to. You have a week off. Please don't travel too far and don't interact with too many people during that time. Keep yourself safe. We'll be back here on Monday the 30th. And I believe Dr. Judd has the lecturing duties for that day. Do you have any homework for them to prepare?
I think actually I have 3 in a row, but you have the first one after the break.
Oh, do I?
I think I've got it right here.
I'm just opening
Review
up
of
Excel.
quantitative genetics and evolution. Oh, and I wanted to remind you all that the first Brody literature summary is going to be December 4th. That's the Friday we get back.
Yeah, and my plan is to get your literature summaries graded and back to you over the holiday so that you have that. So that first literature summary on the 4th is the Friday after we get back, which means that the semester is winding down to a close. We do that one at the beginning so that you then don't have it stacked up on your plate the last week of classes. And it's also one of the simpler of the papers that we're going to be going through. So all of the papers are already loaded onto the Moodle site. My advice would be don't put off reading that first paper until after Thanksgiving. Read it before then so that you can have questions for us when we come back on the 30th. And the other thing that I would advise is after you've read that first paper and think you understand that first paper, start digging into the other papers as well. You're not going to be asked to write literature summaries on them, but you are going to be asked to come to class every day ready to talk about those things in small groups for each day that those papers are on the schedule. And if you leave them until like the night before, you're not going to come into class particularly well prepared for the discussion. Now, the papers that we've chosen kind of walk you through the thought process of what the Brodies did and their collaborators did, but that's primary literature, and you're still getting used to reading primary literature, and that's one of the reasons that we have you reading primary literature a lot in this class. So so don't let those things fall off of your radar. We will also be working on combining the data sets from the two classes. And when we have that done, we'll send that out to you as well from lab and complete with scripts and all that stuff. I just got in the last of the data from some of the groups yesterday. So I've been working through it myself. Dr. Judd and I need to get together and put together a common strategy for analysis. So, any questions?
I have a question about our final.
Yeah.
Could you explain a little bit about how that's going to look like for like exactly what we should be studying more specifically?
So the final as we've done it the last couple of years, I guess, is it's a two-part final. One part is just like a regular exam. It has a short answer section. It has a multiple choice section. And then the second part is actually the pretest that you took. So at the beginning of the semester, you took a pretest. Presumably that is a test that Dr. Judd and I went through a couple of years ago, and we actually revised that pretest such that that pretest is weighted on a day-by-day basis on the material that we've covered across the whole semester. And so presumably the reason that we have a pretest is so that we can give you a post-test to gather kind of what you've gained across the whole semester. And so half of the final exam will be a typical test that is just focused on the last case study. And then there will be a comprehensive section that you take on Socrative that is the post-test. And your pretest, you've got credit for taking the pretest regardless of what your score was. In the post-test, your score is going to be the score that contributes to half of your final. Does that make sense?
And you may recall that that cumulative portion, that post-test, is multiple choice.
Yes, it's on Socrative, it's online, it's multiple choice, it's graded as soon as you finish taking it. So. All right. So yeah, so we've mentioned just in the last couple of days, I've asked you to go back and grab things from your memory banks from earlier in the semester as we start to get ready for this case study. Don't think that just because those other case studies have passed that you can forget that stuff. That stuff will be on the final ultimately. All right. Anything else from you, Dr. Judd?
I don't think so. Just looking on here to double check. If we think of anything else we need to announce to you all, we'll put it in an email or something. But we're going to get these literature summaries and these exams graded. So.
Yep. Dr. Judd has already started grading the exams, and I'm going to start on that today. So we'll get some grades, and we're also I particularly need to get on the posting grades to Moodle, but hopefully here by the beginning of next week we'll have a fully sold out set of grades on Moodle, so you can take a look at that. All right. Well, have a good Thanksgiving. Stay safe. Don't get sick. And enjoy time with your parents. And actually try and take a little bit of a mental break, because I realize that this has been a very unusual semester. It's a Zoom fatigue kind of semester. Trust me, and Dr. Judd, when we say this, we also are fatigued by this. So we know how you feel. So take a breather, recoup, and be ready to come back on November 30th to hunker down and get the rest of the semester finished off.
I did remember the last thing I was going to say was don't forget about your review of the literature summaries. Yeah. But enjoy your break, and we'll see you when we get back.
All right. Bye. Nathan,
Yeah.
I've been working on scripts. Have you been working on scripts or not?
I have a little bit. I've got everything into CSVs. I've been trying to get so a little bit, but not much. I was thinking about it yesterday, but I didn't write a whole lot.
Okay, can we, so let me turn this off.